to FISH500 students presenting. And actually quite cool, they're both from Canada, right? I don't think we have that yet. One announcement, so this is our pre-last talk. We have one more talk next week, which will be Daniel Pauly. So even though the term's kind of over, we decided to just kind of do one additional one. And our first speaker is Clayton. He's a first year's master's student with Amanda Vincent with the project Seahorse. And he's from University of Calgary, where he did his undergrad. So please welcome Clayton.
Everybody, thank you for coming. Hopefully this microphone works. Okay, as conservationists, we're always interested in the sustainability of populations. Whether it be looking at how much of a certain species or fish to harvest in a given year, or looking at the effect of a disease on a subpopulation of a different fish is always the case. When looking at habitat, is no different. But in order for a population to maintain itself through time, it has to be able to reproduce at a rate and produce healthy individuals that overcome the death rate. But this is easier said than done, because the conditions that allow for reproduction are often narrower and less robust than those that allow for survival and growth. And as such, these ecological hoops that the population has to jump through during its life history become narrower and narrower over time. And this is just in one area. But what happens if the population migrates? Well, then it's going to have to overcome these new conditions that might be very foreign to it. Or what happens if external forces change the conditions that a sedentary species experience in one area? Well, we see that every day in the form of climate change. As you can see, this issue is quite complex. But for the most part, all of the focus of research so far has been on the effects of habitat quality on survival alone, and much less has looked at the effect of habitat quality on the sublethal consequences that of growth and reproduction. So that's what I want to look at. And specifically, I want to evaluate the effect that habitat quality has on the growth and diets of marine fish. And then I want to see which biological, physical, and chemical parameters in the world affects things to the greatest degree. And as you may expect, given the fact that there's a giant seahorse on my title slide, and I'm in Project Seahorse, I'm going to be using seahorses as a case study. Yes. Okay, so there are about 48 species of seahorses within the genus Hippocampus, and they range in size from that of your pinky to that of your forearm. And being fairly charismatic, they are a very important flagship species, and were among the first marine fishes included on Appendix 2 of CITES, which states that all importing and exporting nations must show that the sources are legal and that they are not damaging wild populations. In the world, seahorses inhabit various shallow coastal waters, and usually in temperate and tropical climates. And their habitats are usually seagrass beds, mangroves, coral, sponges, macroalgae, and yes. And when I was trying to determine which area to study seahorses, I needed to find an area that had a lot of seahorses because it's an ecological study. One where there is a habitat gradient, so I can look at habitat effects, and good on-the-ground support. And that led me to Brazil, where I'm going to be studying this beautiful creature, Hippocampus radii, the data-deficient, long-snout seahorse. Okay, so Hippocampus radii, like most seahorse species, are exploited in three major ways. The first is in Brazil specifically, is a regulated directed fishery, which takes out seahorses and then uses them and exports them to countries like the United States or also Europe, countries in Europe. And these are used live in aquarium. But this is very, very different from the dry trade, which is supplied mainly by incidental bycatch. This type of seahorse is dried and then used in things like for religious purposes or in traditional medicine. So in Brazil, it is thought that seahorses can cure various ailments from baldness to asthma, gastritis, bronchitis, various wounds, and it's also used in a perfume form to attract the opposite sex. One of the most interesting ways I thought it was used is that it's crushed and then steeped in tea and then used as sort of seahorse tea, which is kind of interesting. But this is very important to this culture. Like a study that evaluated the relative importance of all animals used in traditional Brazilian medicine found Hippocampus radi, my focal species, in 2 states was the second most important and the most important animal of all of them. So this matters a lot. And the third way that seahorses are exploited is through habitat loss, which is much less studied, and we're much less aware of how this matters. So basically, we often know where seahorses live and what habitats they inhabit, but we are less aware of how this matters with respect to population sustainability. And this clouds the ability of conservationists to accurately assess their statuses, and it also prevents them from making appropriate action plans. So by looking at how habitat quality affects their growth and diets, I hope to equip these scientists with the ability to make these more appropriate action plans and the ability to assess populations better, more effectively. So I will be going to Brazil and studying in the northeastern state of Paraíba. And the basic framework of my study is to do a 2x2 factorial design where I'll have 2 habitat types, an estuary and a coral reef. This estuary is the Rio Paraíba right here, and the coral reef exists on the coast. And within the estuary and the coral reef, I want to try to qualify sites that are disturbed and undisturbed. How I will qualify that, I do not know yet. When I arrive in Brazil, I hope to do this. After doing that, within these boxes, I'm going to measure stuff, which is usually what I do, or scientists do. The first thing I'm going to do is get underwater and gain an appreciation of population estimates, so abundance, size structure, age structure, sex ratio, things like that. Then I'm going to do a mark recapture campaign. This has been shown to be fairly effective with seahorses because of their sedentary nature, with high recapture rates in studies on Hippocampus radi in the past. And the last thing I'm going to do is analyze their guts and see what they're eating. And this will be done by anesthetizing seahorses underwater, bring them to the surface, and using a non-lethal method of flushing their guts and then taking that to the lab and analyzing it. So the time frame of my study will be to leave UBC in November, arrive in Brazil, and then study or do fieldwork for about 4 months, and then return in around February. And even in that short time frame, you can see by this growth rate of Hippocampus radi that you can see very large differences and changes in individual sizes, even in a short amount of time, especially for the younger individuals. So I'm very confident that this mark recapture campaign will be effective. And then, okay, sorry, I should also point out that I want to reproduce this growth curve for each of those sites, so the disturbed and undisturbed estuary and the disturbed and undisturbed coral reef, and then relate that back to other parameters that we measure in the field. And things like, I want to measure vegetation because that is related to habitat complexity, and seahorses are found to have higher foraging success in more complex environments. Additionally, the prey that they feed on are found to be in higher abundance in more complex environments. So I want to look at that. Seahorse growth rates have also been shown to be very affected by temperature. Usually a higher temperature to a degree will allow for greater growth. Additionally, seahorses are sedentary ambush predators that use vision, and therefore the light available is also important. And last thing, I'll look at water chemistry. It's not really well known how this affects things, but why not look at it? And since I'm measuring gut contents, I want to see if the gut contents in their stomachs is how that relates to the abundance that exists in nature, and see if they're being selective in some way. We do know that seahorses, as juveniles, are usually more selective and become more generalist over time. I'm going to look at how the feeding patterns change at different sizes. Lastly, although not a lot is known about how or what feeds on the seahorses, there's been recent evidence to suggest things like octopus and large pelagic fish can actually change the abundance quite significantly. So I want to see if they're doing anything, and by measuring them or quantifying them in some way. And then after returning to TBC and plugging away on R for months and pulling out my hair, and then writing for months and pulling out my hair again, I want this information to be fed to conservationists who will then use the information to better assess the vulnerability of seahorses in these habitats, knowing these conditions that exist. And given that I work with the IUCN Specialist Group for Seahorses, which is Project Seahorse, this will be immediately effective. And then, since seahorses do not exist in a vacuum, hopefully some of these results can have larger, can affect or be used for other species and their conservation needs as well. I would like to thank everyone at Project Seahorse, the organizers of FISH500, especially Anna for all she has done, and everybody in my class and all you beautiful people. And lastly, does anybody have any questions?
Thank you, Clayton. We have a few minutes for questions if there's anything burning on anybody's mind.
Oh God.
I'm just wondering, you said you're going to do both mark recapture. Well, I'm a little bit curious how you're going to do the mark recapture first. Are you going to use VIE tags?
Yeah.
Okay. Are you planning on tagging the same individuals that you're going to be doing the anesthetization and the stomach flushing with?
I guess it depends on what exists. If there's a lot of seahorses, maybe I have that flexibility. I imagine I might actually have to do that, where I use the same individuals, because a lot of times in seahorse studies you have a hard time finding them. I really don't know. This is one of the things that it's hard to anticipate until you actually go to the field. But yeah.
If you do find quite a few of them down there, it might be good to do that with the individuals that you have tagged, and then also do similar sized individuals that haven't been tagged, just as sort of a control, and then you can follow it through its life history with the tag, and then compare it to the other similar sized individuals. It'd be interesting.
You mentioned that the juveniles are more specialized in their diet, and then as they grow, they become more like generalists. And that's a pretty unusual pattern. Do you have any idea of why they might do that? And is it just gait size related, or is there something else going on? Usually animals learn how to handle certain items and so on, and as they grow, they become more specialized rather than less specialized.
Seahorse is actually quite unique because as soon as they're born, they're capable of feeding on their own. But it has a lot to do with gape size and snout size. So they're usually the larger prey. You need a larger snout and a larger gape to prey on. But the juveniles are incapable of doing that initially, and then as they grow, they tend to be able to feed on everything. And that's basically, to my knowledge, that's the biggest reason why juveniles act that way.
No more questions? Thank you, Clayton.
everyone.
Sorry. Okay.
Yeah.
Okay, so our next speaker is Vanessa Martin, and Vanessa actually came here from forestry department. She's one of Scott Hinch's students, and I thought it was quite nice. Scott was, I think, our first speaker of the whole FISH500, and Vanessa's the student, our last speaker. Vanessa's a master's student in her second year, and will tell us a little bit about her project. Welcome, Vanessa.
Thank you, Anna. So today I'm going to talk to you a little bit about some preliminary results that are coming out of my master's. Oh, that's good. Oh my goodness. Yes. Oh. There's only 5 buttons and I already pressed the wrong one. Okay, so sockeye are an iconic species in British Columbia. This is largely because of their economic, their ecological, and their cultural importance. And there's been a lot more investment in studying this species in the last couple of decades because their productivity has been decreasing since the early 1990s. They're an anadromous species, and they're also semiparous. And of the Pacific salmon species in BC, they have the highest rate of homing to their natal spawning grounds. And in years where migration conditions are difficult, so for example, the temperatures or the flow in the Fraser River are particularly high, we see really high levels of en route mortality in these adults that are migrating home to spawn. One of the challenges that migratory fishes like salmon face are dams. And as you can see from this photo, with all of the white water here, dams create these turbulent environments downstream in the tail race where water is moving at very high flows. And as you can imagine, it's very difficult as a fish to navigate this area to find the entrance to a fishway, which in this photo would be right here. And swimming in these high flow environments is energetically demanding. And this is particularly a problem for sockeye like other Pacific salmon species because they don't feed during the freshwater migration. So they're operating on a fixed energy budget, and any extra time that they spend in these types of areas can potentially jeopardize their migration success and their ability to reproduce. My research is part of sort of a suite of grad projects that are working on this BC Hydro monitoring project. So we're really interested in the Clayton Dam, which is up here right near the town of Lillooet, about 4 hours north of where we are here. And over the last couple of years, our projects have been looking at the effects of the operations of this particular dam on the ability of 2 stocks of sockeye to make it to their spawning grounds. And the stock that I'm interested in is called the Gates Creek sockeye. And some previous research has shown not only do fish exhibit behaviors that indicate confusion and delay in the area immediately downstream of the dam, but of those individuals who do manage to pass the dam, we see really high levels of post-passage mortality. So many of those fish are actually dying in the lakes immediately upstream. So this is just a simple schematic of the system. In August and September, these sockeye are swimming north through the Fraser, and about 350 kilometers from the mouth, they hang a left into the Clayton River, and this is where they come up against the Clayton Dam. So I'm really interested in understanding what migratory experiences and temperature experiences are associated with being a fish that makes it successfully from Clayton Dam all the way to the spawning grounds at Gates Creek. And to get at these questions, we use radio telemetry. So between 2013 and 2014, with the help of technicians from the St'át'imc First Nation and a whole bunch of people sitting in the 4th row here, we caught and tagged 372 sockeye. And what that meant was we took a radio transmitter with an I-button temperature logger attached, and we put it into the gastric cavity of each of these fish. And these temperature loggers are recording the internal temperature every 15 minutes. So when we take these fish and we bring them back down and release them in the Fraser and make them swim it again, we're getting a record of the temperature water that fish are experiencing. So this is just a map that shows the location of the radio receivers, which are tracking the progress of fish through the system. This is an aerial photograph of the town of Darcy, where Gates Creek enters Anderson Lake, and you can see in the red box there's an artificial spawning channel in the system, which is really beneficial because it makes it significantly easier to recover temperature loggers from dead carcasses, and as well, Fisheries and Ocean Stock Assessment and the Naquaqua First Nation have a really thorough assessment of spawning success for female fish that die in this channel. And I'm going to get to that in a second. So there's over a number of years where we've been looking at the survival of sockeye through Seton and Anderson Lake. We've seen some really interesting trends. These data are from 2007. A previous master's student named David Roscoe found using acoustic telemetry that although 80% of male fish were successful in swimming through these two lakes, fewer than 60% of female fish actually made it successfully. And when we looked at this in 2013, we found like a nearly identical trend, even though our sample sizes were much higher. In 2014, conditions overall were more benign, so temperatures were easier for fish to tolerate, and more fish made it in general, but again, we see that female fish just aren't doing as well. When we look at the amount of time that it takes for a fish to swim from the dam to spawning grounds, so we have number of fish on the y-axis here, the number of days spent in the lakes on the x-axis, and then 3 years of data. We can see that there's huge variation. So some fish are really motivated and they take only 2 or 3 days to swim that distance, whereas others are taking, in 2007, 35 days or 25 days. And this distance is only 55 kilometers. And this is really not a lot for fish. Previous telemetry work that our lab has done in the Fraser River shows that these fish can swim 30 kilometers in a day quite easily. So we know that they're swimming below their capacity, and that just makes me wonder, what are they doing? Are they sightseeing? Are they hanging out? I'm not really sure. So, like other salmonids, sockeye behaviorally thermoregulate. So when they're in environments where multiple water temperatures are available, they tend to be found occupying the temperature that's closest to their optimum. This makes sense for a couple of reasons. It makes their metabolism more efficient, so you can conserve energy, potentially recover from physiological stress. And because sockeye have these natal lakes very close to spawning grounds, there's a potential thermal refuge for adult salmon that are migrating upstream. Like in Seton and Anderson Lake, many other stocks have this place where they can hang out, and they can sort of dictate their own thermal experience. So we put these eye buttons in fish, and between 2013 and 2014, we recovered 113 of them on spawning grounds. So each of these eye buttons has the complete thermal experience of a fish traveling, these roughly 60 kilometers. And when we graph these are two individual temperature profiles. So these are two female sockeye that were released in the Fraser on August 18th, 2014. And although environmental conditions were presumably identical for them, we can see that there's different things going on. They're having different experiences. So I'm just going to take you through that in a little bit more detail. The temperatures in these red boxes are the temperatures associated with the time that the fish spent in Seton Lake, which is highlighted on the map. And I just named them Pamela and Sally because we like to anthropomorphize, but also because it makes it easier. So you can see that Pamela in Seton Lake is spending time around 5 or 6 degrees, whereas Sally is closer to the thermal optimum of about 16 degrees for these fish. And presumably they have just passed Seton Dam, which we know requires anaerobic effort for them to swim through that turbulent tail race environment. They've swum up the fishway. So presumably Seton Lake is the place where they might be recovering from that experience. They might be consuming more oxygen, metabolizing lactate. There's a particular temperature that makes that more effective. Then they pass through Portage Creek, which is sort of the warmest temperatures that these fish experience during this portion of the migration. It's usually 20 or 21 degrees in the summer. And then into Anderson Lake, where again we can see there's kind of a consistency in the behavioral strategies that these two fish are employing. Pamela, again, is in very cool water. Sally at a more moderate or optimal temperature. And Anderson is really like the final stage of the migration for these females. This is where they could be investing in maturing their gonads. They don't really need a lot more energy to be migrating. They can really invest in reproduction. Okay, and then we can see here as well, there's a variation, there's differences in the duration of the migration. So Pamela was about 4 days faster than Sally. So we know that both of these fish are successful migrants. They make it to spawning grounds, but we can take it a step further and we can ask, to what extent do they contribute to the next generation? And traditionally, Fisheries and Oceans has looked at spawning success as these higher level population correlates. So you have a certain proportion of females on spawning grounds who deposit eggs, who are considered successful spawners, and you can relate that to the general environmental conditions that the population experienced during their migration. But using telemetry, we can actually look at the individual migration experience of fish and relate that to their individual spawning success. So we found with Pamela, she failed to deposit any eggs on spawning grounds, and the fish that we looked at in Gates Creek were assessed according to the DFO standards. So it's either 0%, 50%, or 100% spawned. So here we can see in this photo, the skeins are still tight, all of the eggs are retained in the body cavity, whereas for Sally, she deposited nearly all of her eggs. And 46 of the females that we radio tagged between 2013 and 2014 were assessed. So I'm really interested in looking at what might be some of the reasons for failure or success in spawning. And so to summarize, I have a lot of data. Some of the data that I have are information about the initial condition of the fish when we caught it. So this is when it first enters the Seton River. We have information about blood physiology, the injury state. We did a standardized injury assessment for all fish. And I'm interested in whether those factors in initial condition predict survival. So does the fish make it through Seton Lake, Anderson Lake to spawning grounds, and also the migration rate and thermal experience in those two lakes. And then in turn, I want to understand whether initial condition predicts spawning success. Is that predetermined by the time a female gets to this point in her migration, or is that actually mediated by that migration rate or thermal experience, those tactics that Pamela and Sally were employing when they were traveling through the lakes? So I'm going to spend the next year of my life trying to navigate this flow diagram. But in the interim, I would like to thank all of these people who have contributed. It's a really collaborative project. Statlium and Aquaqua First Nations both contributed a lot in the field. We partner with BC Hydro as well as InStream and Limnotech. And Fisheries and Oceans Environmental Watch contributes a lot, and also the Hinge Lab. And then I would like to thank FISH500 and all of you. Thank you.
Thank you, Vanessa. Any questions? Yeah,
It's about the temperature in the lake. Is it possible to have a variation between the layers in the lake and then the salmon to grow increased layers? So that makes it possible that Canada goes after a layer
And lower?
yeah. Lower, and is it these things about the games can affect the salmon or?
Yeah, so one thing that we did, which I didn't talk about here, is we put thermistor chains in the lakes. So we basically strung a line with temperature recorders or temperature loggers at different depths. So I have the information about what temperature the fish was experiencing. I have some information about what temperature the lake was at different depths, so I can try to equate those things. I think it is likely that they're doing these regular vertical migrations. There are other researchers, so Chris Perrin with Limnotech, he does a lot of limnology, and so he's analyzed the water column in both Seton and Anderson lakes, and it doesn't seem like oxygen is a limiting factor, for example. So although there is some stratification, it doesn't seem like there might be negative effects of spending too much time at a deeper layer rather than a shallower layer. But I am interested in why they might be traveling that way. I have, I guess, 113 of those temperature profiles, and some fish show very regular migrations vertically, and some fish really consistently stay at one temperature. And it's hard to know if that's just like a behavioral artifact or if it's actually doing it.
I'm just wondering, when you were on that slide where you opened the fish up to look at their reproductive status, how did you decide when to open them up, and how do you know that the one that hadn't the gonads hadn't matured?
Yeah.
How do you know that it wasn't just delayed and was going to happen a few
Right.
days later?
So in the way that they manage the spawning channel is they let fish in on particular days, and every day technicians go through and just remove fish that are dead. And so they do this as a way of enumerating the number of fish that make it to spawning grounds, like the total population. So they cut all those fish in half and they count them. But for females, they open them up and they assess
Okay,
0,
so they're
50,
dead.
100. So they're dead.
Okay.
Yeah. So that's kind of the only way you can really distinguish between whether, a female is just taking a while. There is, I think, like all of the fish that we presented, they were dead, but there is, once they get to sort of a moribund state, some of them are pretty gnarly, and it doesn't seem like they maybe have enough energy to like dig a red and defend it and deposit eggs and all of those things. So I think there is probably a state beyond which you can say she's not gonna deposit eggs, but yeah, we stick to when they're already dead.
Hey Vanessa, good to awesome talk. Thanks a lot for that. But I know you said you had a ton of data. You didn't really show all those. But you said you had physiological and injury-related stuff.
Yeah.
Are you looking at any correlations with body size and like temperature preferences and
Yeah, I have like fork length for all of them, so I'll probably look at that. I can't think off the top of my head of other studies that have found that to be significant, but yeah, that's definitely something I'll look at. We didn't take a lot of other morphological measurements, but yeah, fork length, we have some indication of the level of energy that fish have, so we have like a energy meter. So yeah, those are some of the things that I know, as well as like reproductive hormones, things like blood, lactate, blood ions.
Well, thank you so much.